Okay, so hi, this is Niladri. This is Niladri. I will be the host of ISCB Students Council webinars today. So today we have a talk by Sania Kular, who is a PhD from University of Wisconsin Madison in USA. She will be talking about ADSNP-Pheno, which is a computational pipeline for predicting regulatory mechanisms in Alzheimer's disease progression. Before we start the talk, I would just like to request everyone to subscribe to the YouTube channel, and like in order to know about the student council webinars, please follow us on on Twitter and Facebook at ISCB-SC. Sania, the floor is all yours.
Thank you. Thank you very much, Niladri. So as Niladri has mentioned, I am Sania Kuller. I am currently a PhD student in the Department of Biostatistics and Medical Informatics at the University of Wisconsin-Madison. I want to thank the ISCB-SC for giving me this forum to present my novel pipeline, ADSNPheno, a computational pipeline for predicting regulatory mechanisms in Alzheimer's disease progression. Alzheimer's disease is a form of dementia with memory loss and cognitive disability with no known cure. We typically look at the AD progression from the initial to the severe stages: psychological resilience, apogenotype, Braak staging, CRED score for neuritic plaques, and cognitive impairment based on a mini mental state exam. Braak stages 1 to 6 help explain the neuropathology of AD by focusing on the accumulation of neurofibrillary tangles and the spread throughout the brain where they disintegrate the microtubules, disrupting neuronal transport and functioning, leading to neuron death and dementia. SNPs, or single nucleotide polymorphisms, at one location DNA base changing, are the most common type of genetic variation. Many SNPs and protein coding DNA have been found. We know about 30 different risk loci for late onset AD. Unfortunately, apart for ApoE4, which you see here is really high, there are no causal determinants that have been found for late onset AD. Well, why is that the case? Well, so far, we have not really looked into the role of SNPs that occur in non-coding regions in AD. This is where our pipeline comes in. A lot of research has been focused on the 10% of SNPs, those that occur only in protein coding regions, although around 98.5% of the human genome is actually non-protein coding. In fact, around 90% of SNPs occur in non-coding parts of the genome, such as transcription factor binding sites. So, what is the functionality of SNPs in these non-coding regions in AD progression? Well, for that, we need to understand how these SNPs in non-coding regions can impact the regulation of genes involved in AD, because SNPs can also be useful even if they are not in protein coding regions. So here I have honed in on an important AD progression transcription factor, REST, whose binding to this transcription factor binding site is disrupted by this SNP. This SNP is given by this ID over here on chromosome 19. We see this SNP here. It changes this base from a C to an A, and REST's ability to bind to this transcription factor binding site is impacted. And I'm going to explain that more next. I'm going to tell you guys what exactly are these transcription factors. Well, transcription factors help regulate the expression of genes, the activation or the repression. TFs bind to DNA sequence-specific transcription factor binding sites near the enhancer or promoter of the target gene. So here what we see is that there are all these different factors here that are involved in helping recruit RNA polymerase II to transcribe the gene into mRNA and then ultimately into protein. But here we see these transcription factors are binding to the enhancers and the promoters, and then they are involved in helping recruit RNA polymerase II. And these are specific proteins, and they have sequence-specific motifs. So here, this transcription factor, for example, is TACGTAC, and it binds to the complementary sequence ATGCATG, much like puzzle pieces. And by binding to the transcription factor binding site, it helps with the regulation of this gene. So we see here, for instance, that even if one base were changed, it would impact potentially the ability of the transcription factor to bind there. And that's why like these SNPs here can potentially impact the ability of transcription factors to regulate those target genes. So research is being done on the effects of SNPs disrupting TF affinity to TF binding sites. For example, SNP to TFBS is a database dedicated to exploring these effects. ADSNP is another software. The paper on the right has found SNPs that disrupt TF binding sites, which lead to an increased risk of schizophrenia. Functional genomics looks at AD variants and tries to understand how those relate to AD risk genes. How do genes control one another and function throughout AD progression? Even in this highly regarded paper on functional genomics for AD progression, the authors have only identified AD risk SNPs in protein coding regions. Their work has been a very valuable contribution, but still there is another area that remains largely unexplored. And that is where our pipeline steps in. In our novel computational pipeline, we take a functional genomics approach to understand the role of SNP variants in the regulatory regions like transcription factor binding sites. This is to help us find potential underlying causal mechanisms of late onset AD in different major regions of the brain. Because we still do not have an answer for that. We are hopeful and confident that our pipeline can help us get there and also provide a framework for researchers for other diseases as well. Through functional genomics, we can output AD regulatory networks linking SNPs, transcription factors, and regulatory elements such as enhancers to target genes for AD phenotypes such as AD progression stages. So, we have come up with ADSNPheno to predict regulatory networks for AD progression. We have applied it to major AD-related brain regions and tried to predict gene regulatory networks and functional genomics for AD progression phenotypes. And we have been building an interactive functional genomics resource for AD progression. So as I mentioned, our understanding of the cellular and molecular mechanisms from disease risk variants to phenotypes in AD is still limited, especially during AD progression. And again, a majority of the SNPs are on non-coding regions. And it is a bit more difficult to link them to genes and then to functions. So we need some approaches to link these non-coding SNPs to genes. Our approach is to use gene regulatory networks to link SNPs to genes. While this methodology has been applied in other contexts, ADSNPheno is focused on AD progression in 3 major regions of the brain. Our computational pipeline, ADSNPheno, helps uncover gene regulatory networks from AD risk genetic variants, such as SNPs, to phenotypes. So ADSNPheno inputs multi-omics data, and then we create and construct a gene co-expression network using the gene expression data to come up with gene modules. We also come up with gene regulatory networks for AD phenotypes, like progression stages, and then we associate those gene modules with AD developmental phenotypes. And we find enriched functions and pathways for those gene modules. Then we also look at SNPs breaking transcription factor binding sites and identify transcription factors, enhancers, and genes. And then we tie all this information up into our output, gene regulatory mechanisms for AD progression, where we look at the SNPs that are impacting transcription factors and transcription factor binding sites, which impacts the ability of transcription factors to bind to these and regulate these enhancers. And because of that, the transcription factors are unable to properly regulate these genes. And then we look at these genes and the gene modules they belong to, and what functions and pathways they are involved in, and what AD phenotypes they are associated with. So here we can go from SNPs in non-coding regions all the way to AD phenotypes. So we can go from SNPs and non-coding regions to AD phenotypes again. And that is SNPs to pheno, AD-SNPheno. So we focus on these three AD-related brain regions: the hippocampus, the lateral temporal lobe, and the dorsolateral prefrontal cortex, which has a majority of our samples. Weighted gene co-expression network analysis with k-means was applied for each of these three brain regions. And essentially, we take this gene expression data and we group it into genes that have the highest similarity or co-expression, the strongest connections with one another based on correlations. Each of our gene module had at least 30 different genes in them. So here, with ADSNPheno, for each of these three brain regions, the hippocampus, lateral temporal lobe, and dorsolateral prefrontal cortex, we built gene co-expression networks for each of them. And the gene modules are this tight group of genes that have very, very, very tight, very well connected. And for instance, and then once we have these gene modules, we can see these, functions that they're involved in, hub genes very tightly connected, pathways and enrichments, and phenotypes that they are associated with, as I will explain soon. But for instance, the hippocampus had 31 human brain samples, 7 control and 24 in various AD stages, such as initial, moderate, and severe stages, and led to 30 gene co-expression modules for various AD phenotypes, including staging. For now, I will focus on the application of ADSNPheno to the lateral temporal lobe, where we created 56 different gene co-expression modules. So here we are associating modules with 80 developmental phenotypes in the lateral temporal lobe. And genes in different gene modules show different gene expression dynamics during AD progression. So here we can focus on, for instance, if you're having Alzheimer's disease or not. So orange is if you have Alzheimer's disease, and yellow is if you do not. So this heat map here is showing, how these represented modular expression patterns, also known as eigengenes, vary for AD phenotypes. So ME stands for module eigengene. And for instance, this magenta module has a high expression for individuals who have Alzheimer's disease. So we see here for expression, when it's red, it's higher expression, and as we go down to blue, it's lower expression. So red here for those who have Alzheimer's disease, and it's lower for those who do not have Alzheimer's disease. So for these individuals. So that's what we see here, that this magenta module typically has high expression in individuals with Alzheimer's disease and lower expression in those who do not have it. Using these expression dynamic patterns, we further linked modules to all key phenotypes, including AD staging. This Christmas-themed heat map shows the corresponding correlation R values on top and the respective P values below in each cell for modules that are strongly associated with key phenotypes. Significant associations are those where the P value is less than 0.05. We focus on the positive correlations, that is for R greater than 0. So let's hone in on this module right now, again. So for instance, we see that this magenta module is more strongly associated with having Alzheimer's because it has an R equals 0.51 and a P value of 0.004, which is less than 0.05. So it is really significant and strong. So there's a significant strong association between the magenta lateral temporal lobe and having Alzheimer's disease. And it's more significant than this midnight blue module, which only has a correlation of 0.31 and a p-value of 0.1, which is not significant. So we can zoom in some more on this magenta module and see a more detailed view of these different phenotypes that it is correlated with. We can observe that it has a strong positive correlation with having Alzheimer's disease and with dementia, BRCA 6 stages, BRCA 5, having more of these plaques, worse CRID score. So overall, I would say that this magenta module and the lateral temporal lobe is strongly positively associated with AD progression. But it's also not just strongly associated with AD progression, but it is also biologically meaningful. So we've used hundreds of different sources to gather enrichment information for each gene module, such as this magenta module. And we are presenting some of the key enrichments here. For instance, this magenta module is enriched for the stress-activated and TRAF6 mediated NFKB signaling that phosphorylates the IKB molecule, which is associated with AD. It's also enriched for the I-kappa B kinase complex, death receptor signaling, negative regulation of gene silencing, and a positive regulation of intrinsic apoptotic signaling pathway in response to DNA damage. Those are some pretty bad phenotypes, enrichments that it's associated with. So as we see here, an AD, calcium dysregulation is a factor in AD. In this slide, KCNN4 is a calcium-activated potassium channel gene, and it is our target gene. And the lateral temporal lobe, it is regulated by these two transcription factors, REST and BCL6B. KCNN4 is typically expressed at low levels in healthy neurons and microglia, but it is overexpressed in the AD brain. Studies have confirmed that by blocking KCNN4, we can decrease neuronal loss and improve memory levels over time. So KCNN4 is a gene in the magenta AD progression module, which, as we have seen, has high positive correlations with having AD, with the advanced BROC stages like BROC5 and BROC6, with having a worse CRID score with more neuritic plaques, with more dementia overall. And we saw that it has biological enrichments with horrible pathways that are implicated in Alzheimer's disease. So, what are some possible reasons why KCNN4 may be overexpressed in the lateral temporal lobe? Perhaps an explanation could be the dysregulation of KCNN4 by its transcription factors. We can look into our list of AD SNPs in AD patients and see if any of those can disrupt the TF regulation of KCNN4. We know that REST is a key transcriptional repressor in AD progression. It helps modulate neuron differentiation and neurogenesis. It represses genes that promote cell death in AD, and it protects neurons from oxidative stress and plaques. However, recent studies have found that in AD, REST is unable to bind to chromatin and cannot repress its target genes properly. Could it be that REST regulation of KCNN4 is impacted by any AD SNPs? Let's see. And indeed, through our pipeline ADSNPheno, we have found that in the lateral temporal lobe, there are two different SNPs that are directly on the enhancer of KCNN4. Both of these SNPs disrupt the ability of REST to bind to the enhancer and properly repress KCNN4. And again, this overexpressed KCNN4 gene belongs to this magenta AD progression module in the AD brain region of the lateral temporal lobe, which is correlated with worse AD phenotypes, which we have just analyzed. So again, with these disrupting REST binding to the enhancer, KCNN4 expression is increased. So why is this important? We have indeed found that REST repression of KCNN4 in the lateral temporal lobe is disrupted during AD, which probably contributes to the increase in KCNN4 that is found in AD patients. So from here, I have finished demonstrating an application of our pipeline to the lateral temporal lobe and how we can uncover some stories behind some key changes in AD progression in phenotypes that we observe. We applied it to traditional regions like the hippocampus and the dorsolateral prefrontal cortex. And we found that there are a lot more AD SNPs that impact REST regulation of KCNN4 in the hippocampus as well. So I have finished showing you a full application of ADSNPheno. We are very fortunate to have come upon so many interesting findings like these through our pipeline. So I highlighted the magenta module in the lateral temporal lobe with all of you. But I also want to share an important module that I found in the dorsolateral prefrontal cortex using our pipeline. This is the TAN gene module. It has 1,199 genes and is highly correlated with important AD phenotypes. Many of the genes in this module are implicated in the COVID-19 disease pathway, as we will soon see from the enrichment results. I believe that understanding COVID-19 might help us better understand AD and vice versa. I want to explain why. Studies have found that COVID-19 may even increase a person's risk for Alzheimer's, Parkinson's, and other brain disorders. Researchers are trying to investigate this correlation between coronavirus and Alzheimer's disease. Well, what is the connection between them? The neuroinflammation that is found in COVID-19 patients and AD patients may be caused by similar genetic mechanisms. Both COVID-19 and AD are associated with a dysregulation of the innate immune system response. So the hub gene, the most important well-connected gene in this TAN module, is TMEM123 that helps with mediating cell death. And during AD, there is a lot of neuronal death and cell death and a decrease in neurogenesis. So this may be important. This module has significant positive correlations with key AD progression and cognitive decline phenotypes, as we see here, like aging, later onset of AD, dementia, cognitive decline. Patients with the A4E4 alleles for the APO gene also have an increase in the expression of the TAN module gene. That is, like if you have these two alleles of this key APO gene, the huge risk factor for late onset AD, then you have an increase in the expression of these genes. And in fact, if you have ApoE4, unfortunately, you have a 15 times greater risk of having Alzheimer's disease, unfortunately. But sadly, it doesn't just end there. If you have ApoE4, you're also not just likelier of having such a high risk of having AD, but you also have a higher risk of testing positive for COVID-19 and having severe COVID effects. For instance, these articles are saying that risk of severe COVID-19 infection was reported to be increased in E4 carriers, that they are twice as likely to test positive for COVID-19. And individuals and researchers are looking into biological factors linking ApoE4 variant and severe COVID-19. So again, this is really horrible. And this TAN gene module helps illustrate the connection between having ApoE4 and having COVID-19. As we can see here, the key enrichments for this TAN module include the COVID-19 adverse outcome pathway and coronavirus disease, COVID-19. And they're very highly significant here because I've only shown some of these significant enrichments for this module. Some of these other key enrichments include immune diseases like Wegener's granulomatosis and neuroinflammation, HIV infection, neuron death, and Alzheimer's disease. There are many, many horrible effects associated with this gene module. And indeed, I believe there can be a possible link between Alzheimer's disease and COVID-19. The NIH is trying to track down neurological symptoms found with AD because unfortunately, 4 out of 5 individuals who are hospitalized with COVID we've been finding that they typically report neurological symptoms even months after recovery. So here, I was trying to analyze the role of the TAN dorsolateral prefrontal cortex module genes in this coronavirus pathway in KEG. There are many parts that it's involved in, but I want to hone in on a key pathway over here of COVID-19. And these gray shadings represent parts of the COVID-19 pathway that the TAN module genes are associated with. So it's quite a few. And then I was also wondering, well, why is it that ApoE4 is also associated with such horrible COVID-19 outcomes? So I was looking at these correlations, and this red shading here, is for the C1QRS complex 1 of the COVID-19 pathway. And it indicates that the TAN module genes that are involved in C1QRS complex are positively correlated with having ApoE4. And this is sort of where this complement receptor 1 is. So, therefore, I think the added risk that ApoE4E4 can have on the COVID 19 severity may be associated in some way with C1QRS complex. More investigation will be needed, but this is some analysis from our pipeline. All in all, ADSNPheno reveals gene regulatory mechanisms from Alzheimer's disease, AD variants, to genotypes. ADSNPheno identifies the gene co-expression modules for various AD phenotypes via clustering gene co-expression networks. It predicts the transcription factors that regulate the genes in each module, as well as the AD SNPs interrupting the transcription factor binding sites on the regulatory elements. ADSNPheno constructs a full gene regulatory network that links SNPs, transcription factors, and regulatory elements to target genes for each phenotype. This network thus provides mechanistic insights of gene regulation from disease risk variants to the phenotypes in AD. This screenshot of our web tool illustrates how we can visualize a subnetwork for a given gene in a given region of the brain. Here, we analyze the SNPs that impact and disrupt the regulation of the BMP4 gene in the hippocampus. It is a key gene that is involved in neurogenesis, in the creation of new neurons in the hippocampus. There are many different metrics that we can visualize as well, such as the association of that gene with various phenotypes and the association of its module with various phenotypes as well. And as we see here, the same subnetwork for BMP4, we can select on the hippocampus and region and this target gene, get some information on this gene and the subnetwork here. And we are building a really comprehensive data resource as well with lots of enrichments, lots of information, so it can be a functional genomics resource for AD progression in these major AD brain regions. And we hope it will be very helpful and interactive for all of you. We hope that this web tool can help the research community better understand Alzheimer's disease. Overall, we have tried to understand the functional genomics and gene regulation to help explain the relationship between genotype to AD progression phenotypes better. And we want to provide an open source framework so that all of you can better understand genotype to phenotype links for your diseases and your research. In the future, we have some more projects in mind. We want to identify the most important SNPs in AD progression. We want to do single-cell data integration, relate genetic variants to brain development, and use machine learning for predicting AD progression. Huge thank you to Professor Dai-Fen Wang for his amazing mentorship throughout, to Jonathan for his assistance on this project, and to my family and my friends for their support. We thank the supports by the NIH and NIA and the Department of Biostatistics and Medical Informatics at the University of Wisconsin-Madison, as well as the Weizmann Center. I am currently working as a PhD student in Professor Dai-Fen Wang's laboratory at UW-Madison, where we use machine learning to help advance knowledge of human development, developmental disabilities, and neurodegenerative diseases. I will also be putting up helpful tutorials on YouTube related to my research. I hope it can help all of you as well. Thank you so much for listening to my webinar today. If you have any questions, feedback, or ideas, please note that you can message in the chat right now, or you can email me directly at scutter2@wisc.edu. And you are welcome to visit our lab website with Professor Dai-Fen Wang at daifenwanglab.org. Thank you so much.
Okay, great. It was a really nice talk. I can only clap. I can't make other people clap. So I was really amazed by the amount of work that was there, and it was put very nicely. So great job. I mean. I am unsure if there are any doubts over here now. In case anyone has any doubts, as and when people see the video, please get in contact with her. Her email ID is over there. I just wanted to ask, you only looked at the cases where there was a positive correlation, Was there a reason why the ones that had a negative correlation was not taken into consideration? Because I remember from your heat map, there were cases which are both positively and negatively correlated. I might have not understood that part, might be so.
Yeah, so that's a very good question, Niladvi. So we were focused on the positive correlations because we wanted to see what was associated with dysregulation with an increase in expression usually. Well, what is that associated with? So a lot of times for some of the phenotypes that we wanted to see where there was a gain in certain functionalities that occurred because of AD dysregulation. So many of the phenotypes I encoded as the opposite as well, so it was easier to focus on the positive correlations that way. One of our phenotypes was like points that you've earned on a mini mental state examination. So the higher you perform on that, the better you are doing in terms of your cognitive abilities. So I encoded the opposite, which is showing a decline in mini mental state exam as a phenotype. So in that way, if the more points you lose on it, the greater the cognitive decline. So for a lot of the phenotypes, I also encoded them in reverse if possible to demonstrate cognitive decline. And then it was easier to focus on the increase in expression of this gene is responsible for the decrease in this. And in some ways, I focused only on the positives, but for the phenotypes I thought were very helpful, I encoded them in the opposite direction as well. So we have like an increase in mini mental state exam scores, and then we also have a points lost on mini mental state exam. And then if there's a positive correlation with that, then it shows that there's a positive correlation overall with cognitive decline.
Okay.
Does that make sense?
Yeah, it does make sense. It was nice. It's great. Thanks a lot for answering that. Okay, so best of luck with your future work. With that, I will say bye. Thanks a lot for presenting.
Thank you so much, Nalaji.
Okay, bye bye.
Bye.